I think we're going to get started. Welcome, everyone. My name is Greg Tilka. I'm a faculty member in plant pathology and microbiology, and today's presenter is Jensen, who has worked in my lab for the past several years. I'm kind of going from memory on some of your background. So you were originally born in Iowa, right?
Nope, Arizona.
You were? Your parents are originally from Iowa,
Pure
right?
Arizona.
Oh, you're pure Arizonan, okay.
We won't
Continue on that line. So he got his bachelor's degree and his master's degree at the University of Arizona. And during that time, he also did a couple internships with industry. And then he decided to come to the natural beauty of, oh, you took your picture away, the natural beauty of Iowa compared to Arizona. And so I can understand why he chose Iowa.
I know.
He came here in January of 2014 to work on a PhD. He's done great work. He's planning on graduating this December. And without taking up any more of his time, I'm going to turn it over to Jared.
Awesome, thanks. Well, today I'm going to talk to you guys about my dissertation research project, and we're looking at the effects of nematicidal seed treatment on the biology of soybean cyst nematode. Kind of give you a roadmap of how this presentation is going down. First, talk a little bit of background on the soybean cyst nematode, then about some nematicidal seed treatments, and then my work on developing methods to study nematode biology, and then lastly, using those methods to study these nematicidal seed treatments. So the soybean cyst nematode has its origins in Asia, was first discovered in the US in North Carolina in the 1950s, and has kind of spread over to the Boot Hill of Missouri area, and since then to almost every soybean growing region in the US and Canada. It's the most damaging pathogen of the soybean in the US, causing over $1 billion in yield loss annually. In 2014, that equated about 129 million bushels lost. And it causes about 30% yield loss with no symptoms or that farmers can see. So this is kind of one of the big reasons why it's such a problem. A lot of people don't know they have the problem. The symptoms for an SCN infection in your field aren't necessarily specific to the nematode itself. They can be confused with other diseases in the field. You get signs like stunting, mid-season yellowing, early maturation of the plant, decreased root mass and root rhizobia, all leading to yield loss. The only sure-tell way that you can use to tell that you have nematodes in your field is by digging up roots and looking for these small white females on the roots of your soybean plants, or taking a soil sample and looking for cysts or eggs within the soil. So these are what the cysts would look like. So as any good plant pathologist, we have to have our life cycle. So here I'm going to start out at the egg. Within the egg, the embryo develops into the first stage juvenile called the J1. Here's kind of what it looks like, nice little jelly bean. Within the egg, the J1 molts into the second stage juvenile, the J2. And the J2 will hatch from the egg in response to environmental cues. And kind of one of the interesting things about these nematodes is not all the eggs will hatch at once. So it'll be dependent on time, temperature, moisture, and presence of a host. Moving on, once the nematode hatches and it's going through the soil, it moves via water films on soil particles until it finds a soybean root to infect. This is what our little guy looks like. Using its stylet, which is like a needle-like tooth, it pierces into root cells and moves within those cells until it gets to the vascular cylinder of the plant. From there, it chooses a feeding site to start feeding. And so these are staying nematodes within a root, all kind of feeding around that vascular cylinder. Once inside the root, the feeding J2 secretes effectors or proteins into this feeding site to turn it into a syncytium, a specialized feeding site for the soybean cyst nematode. Here we have biochemical changes and metabolical changes to that feeding site that allow that to become such a nutrient sink, enabling the developing juvenile to get larger and larger and steal more from the plant. So this is what it looks like with the nematode getting larger and larger within the plant. Here, as the juvenile feeds, it molts again into the third stage juvenile, and once again into the fourth stage juvenile. The fourth stage juvenile males will revert to worm shape, kind of tucked within this cuticle like you see here, and the females become so large, their posterior end is exposed on the outside of the root. Finally, the juveniles will molt once more into the adult stages. Males will leave that cuticle entirely and look for females to mate with. Males are much larger than the J2 counterpart, and these guys don't feed. Females become that lemon cyst shape that we know is pretty stereotypical of the soybean cyst nematode. Males will mate with multiple females, and females will be mated by multiple males. Once females are mated with, they fill up with eggs all outside of the root. And once their body fills up with so many eggs, the females die, and their bodies fall off their roots and harden into what we know as the cyst, this brown cyst, and that protects the eggs. And eggs within here can last for up to 10 years. Sexual reproduction between the nematodes leads to high genetic variability, which helps the nematode kind of evade a lot of stresses down the way. Each female fills up with about 200 to 300 eggs. You get a ton of eggs per female, tons of females per root, tons of generations per growing season, and that's kind of where our snowballing effect of that massive soybean cyst nematode problem we have. Get about 3 to 7 generations per growing season, and again, the eggs can live for up to 10 years within that cyst, or stay dormant. So management of soybean cyst nematode is kind of divided into two ways we can do it. First is we can grow a non-host crop. So in Iowa, that only means corn. And with that, when you plant a non-host crop, some of the eggs will hatch because of timing and moisture and environmental cues. And they'll try and go and feed on the corn roots, and they're not going to be able to develop a compatible feeding site, and they'll die. But because not all the eggs hatch, there's still going to be some nematodes left within the soil. So growing non-host works, but it doesn't kill all the nematode eggs. The next option is growing resistant soybean varieties. So a combination of different resistance genes confers resistance. There's about 900 available varieties in Iowa to the farmer. However, only two sources of resistance are bred into those 900 varieties. About 3% is peaking, and about 97% is PI88788. And these are just lines that were found in the plant introduction lines back in the 60s when the soybean cyst nematode was first discovered. So prolonged use of PI88788 has led to the soybean cyst nematode in the field able to overcome a lot of this resistance. And these plants aren't being as effective anymore. And it's kind of like if you were to grow or use glyphosate over and over and over again, you're going to see a lot of resistant weeds. That's why I see a water hemp field with a couple of soybean weeds grown in it when I drive here. So this all kind of leads to the new need for SCN management options. And kind of sticking with the newness, let's talk about some seed treatments. Seed treatments are kind of a newer option in nematode management. We have quite a few treatments out on the market, or coming out onto the market now, either chemically based or biologically based, that all have a different mechanism of action, hopefully reducing the number of nematodes early on in the season, and hopefully trying to help your plant get a better stand and be stronger later in the season. The two treatments that I'm going to be talking today about are Evicta and Chloriva. They're both produced by Syngenta. So Evicta, with its active ingredient abamectin, is kind of an old chemistry. It's also known or used to control animal parasitic nematodes like heartworm in dogs. It's a general nematicide seed treatment used on corn, cotton, and soybeans with effectiveness against root knot nematode and reniform nematode. It's the fermentation product of a soil bacteria Streptomyces avermitilis, and it causes a cellular ion imbalance leading to paralysis in the infected nematodes. It's used as a seed treatment in this case because it has a high affinity for soil particles and organic matter and fairly insoluble in water. The second treatment is Chloriva with its active ingredient Pasteuria nishizali. This bacteria is a gram-positive endospore-forming bacteria, and it's an obligate parasite of the soybean cyst nematode. How it works is as the nematodes are moving through the soil, spores will attach to the cuticle. Once the nematode enters into the root and starts feeding, a germ tube from the spore attached to the cuticle moves into the nematode body. And within there, as the nematode continues to feed, colonies of spores of this bacteria start to proliferate throughout the body, eventually killing the nematode, releasing more spores out to the environment. So the roadmap for my dissertation project was kind of twofold. First was to develop new methods to study nematode biology in the lab. And then the second was to use those new methods, plus some established methods, to study how the effects of these seed treatments affect the nematode biology. The players that I'm working with in here are two varieties of soybean seeds, Williams 82, which is an SCN susceptible seed, and Jack, which is a resistant seed coming from PI 88788 resistance lineage. And both of these are public soybean varieties. So these two treatments, plus the two seed treatments, Avicta and Clariva. So the goal, my overall kind of goal with this, was to isolate out life stages and processes and to determine how these seed treatments affect that specific aspect of biology. And this was done with lab and greenhouse experiments. So let's first talk about the egg. What can we do about the egg? We can look to see, do seed treatments affect SCN hatch rate? So the method we came up with was incubating eggs on a 30 micrometer pore sieve in a treatment solution. And then with that, you would transfer it at 3, 7, and 14 days. Just using a simple 6-well plate, you get 2 replicates per plate. And then after each transfer, you count the number of hatched juveniles. And then at the end of 14 days, you count the number of hatched juveniles and unhatched eggs. The treatments I used were soil leachates that I collected by planting a treated seed, about 6.5 grams of sterile soil in a microsieve, adding about 2 mils of water and incubating it for 24 hours. From there, I would flush 2.5 mils more of water through and collect a leachate, kind of combine it together, and use that for subsequent experiments. So these soil leachates will kind of come up and up and up again throughout this presentation. The treatments I used for these leachates are Williams 82 with the Avicta treatment, Williams 82 with Clariva treatment, untreated Williams 82, and fallow or unplanted soil. So with our treatments, I also have a couple control treatments. So Avicta solution, so what was put on the seeds, Clariva solution, water, and a positive control of zinc sulfate. And then this graph, all we're looking at is the number of hatched juveniles over time. The big thing that pops out first is that we have this massive increase in hatch of this blue line, which is our positive control, meaning, hey, our experiment worked. The next thing we want to look at is what didn't work, right? What stopped the nematodes from hatching? So looking at these two shortest lines, that relates to Williams 82 with Avicta and our Avicta solution. Jumping into the stats of this, I wanted to look at the cumulative percent hatched. So this is just the total number of juveniles hatched divided by the total number of juveniles hatched plus unhatched eggs. And so we're looking at this at the end of the 14 days. And again, we see this high bar related to our zinc sulfate and these two short bars of our Avicta treatment and our Avicta solution, kind of showing us that Avicta is a pretty good hatch inhibitor. All right, so once the juveniles hatch from the seeds, let's move on to figure out how we can attack the nematodes at probably its most vulnerable stage, when it's outside the egg and before it's in the plant. One of the things we want to look at is, do seed treatments affect the movement of nematodes, perhaps preventing them from reaching the root? So one of the things that I kind of struggled with was a lot of methods that looking at the effectiveness of seed treatments, just incubate nematodes in treatment and look at them under a microscope. And then someone will count, okay, I see 12 moving and 5 not moving. So here's a video that's kind of washed out, but there's nematodes on here. They're all kind of bunched together. And if one person were to look at that at one specific time, they might see a certain percentage of nematodes that are moving. And other person might see a different percentage. And that's not what we want. We want something that gives us much more concrete data. So working with an electrical engineering lab, we came up with 2 methods to study movement using computer analysis at the population and individual nematode level. So using time-lapse imagery with computer analysis, it helps give us qualitative or quantitative data, which is much stronger than just saying, hey, this nematode's dead or alive. The treatments I used in both of these methods were soil leachates, so our Avicta seed treatment, Clariva seed treatment, untreated and fallow, with our control solutions of Avicta, Clariva, a water treatment, and heat-killed juveniles. Active juveniles were incubated in one of these treatments for about 2 hours at 25 degrees. I obtained active juveniles by placing freshly hatched juveniles, again on just that 30 micrometer pore sieve. Juveniles that are active move through these small pore openings into water below, and I just collect those after about 3 hours. Once all these nematodes have been incubated in those treatments, that treatment solution gets really dirty, and you cannot take video images on the scope of that. I wasted about a good 6 months trying to figure that one out. So what I found was if we use sucrocentrifugation, we can recover the nematodes from the solution. They're still been affected, and it's a nice clean solution that we can take an image with. So the first is looking at juvenile movement of population. This employs a high-res flatbed scanner using 24 well plates filled with a chloronic gel, which is just a very clear media. And using computer analysis, we can output percent movement of a population. And this is done with help with Augie Beeman, who I graduated last spring. And so with this method, we can simultaneously monitor multiple nematodes per replication, multiple replications per experimental run, and multiple time points throughout the course of a day, all in a semi-automatic manner. So in this graph, I'm showing you kind of figure two treatments. An untreated, so just nematodes incubated in water, and a heat kill treatment, so dead nematodes. At time 0 hours, when we place the nematodes in these wells, these green circles indicate an individual nematode. They're both they're both centrally located where we put them in. But after 24 hours, blue circles indicate nematodes that had moved or dispersed from that input port, and red circles mean that those nematodes had not moved. Over 24 hours, we can see quite a bit of dispersion of the nematodes incubated in water, whereas the heat kill nematodes have not moved at all. So kind of proof of concept, hey, this works great. Let's take it one step further. Next, I wanted to look at abamectin, which is just, again, a common nematicide. And just a reminder, it's the active ingredient of Avinta. And we looked at 5 different concentrations, 0, 0.1, 1, 10, and 100 milligrams per liter. And all we're looking at is the percent movement of a population over 24 hours. First thing we see is a fairly big divide between 2 treatments and 3 treatments. And that's kind of the big take-home point here. Higher abamectin concentrations, 1, 10, 100 milligrams per liter, have shown that they inhibit nematode movement. And we get abamectin is as effective as low as 1 milligram per liter. So this was another kind of validation experiment of this method to show that it worked. And then from here, still kind of processing data from my soil leachates, and that should be analyzed soon. So next, let's look at juvenile movement of the individual. Using overhead light microscopy with a high-speed camera, we can take images of nematodes every 1/10 of a second at 63x magnification for 1 minute. Converting it to binary colors, that allows the computer to identify the nematode with a black background and add 13 evenly spaced tracking points along the nematode body. Each of those tracking points records an XY coordinate at every 1/10 of a second for 60 seconds. So using those tracking points and those XY coordinates, we can quantitatively measure multiple movement parameters of these individual nematodes and see how they respond to these different seed treatments. And with this method, we're able to measure microscopic changes in movement, so we can kind of get an idea of if we see a lethal effect or even some sublethal effects. So kind of diving into that a little bit further. No, just kidding. Let's talk about the parameters calculated. The first parameter is speed, and this is just the change in distance of a tracking point between one time observation and the next. The next is curvature, and this is a little bit complex. This, using the measure curvature method, measures the radius of a circle of a given point of interest that is tangent to 2 lines coming out of that point of interest. The larger the radius means the larger the curve. So say we want to look at the radius at point B. What we would do is we would look at the radius of a circle that's tangent to lines AB and BC. This radius is a function of that angle or that curve at that point. And again, the larger the radius, the larger the curve. The radius at point F is much smaller than at point B, so we would say this is a larger curve. In my data, we're measuring the change in curvature, so that's the change in the radius length between time points. So if we look at just one tracking point, the head, we can plot the speed of that tracking point over time. So this is up to 600 milliseconds. So every 10th of a second, we have a plot. And we can compare tracking head of treatment 1 versus that of treatment 2 and look at some of the trends. Treatment 2 is much slower than treatment 1, but it's still not at 0. If you were to look at that under a microscope and try and say, did this treatment work or not, you're still going to say, no, this treatment didn't work. But clearly, it had some sort of effect. And this is where we can measure those sublethal effects that these treatments may have on the nematode. All right, so onto my soil leachate data for this. We use 3 equidistant tracking points, the head point, the midpoint, and the tail, to kind of give us a whole understanding of the nematode movement, because it would be ridiculous to analyze all 13 points. Looking at this graph, we're just plotting the speed in micrometers per second for our different treatments. And in the black bar, we're just looking at the head. And what's interesting to point out here is that our 2C treatments, Williams 82 with the Victa, Williams 82 with Chloriva, had significantly slower movement than that of our water control. So our C treatment soil leachates do slow down the head compared to our water control. We see similar trends for the midpoint and for the tail. Midpoint and tail follow similar trends. Tail's a little bit slower than the head. And you're probably looking over here and you're like, what in the hell is this big bar right here? So I got a little video to show you. These are our tracking points, and this is what the video looks like when we're tracking a nematode up close. The front, or the anterior and posterior are fairly immobile, but these inside little ones, they keep vibrating back and forth. So it's going to pick up a little bit of that change in speed right there. So how we combat this is we look at another movement parameter, and that's change in curvature. So if we look at change in curvature, that's just 1 over micrometers, which again is just the change in that radius length from time point to time point. For our different C treatments and our control solutions, and again, the black bar just looking at the head. A couple of things we see here. A Victa C treatment, Chloriva C treatment, and a Victa solution have shown slower movements or changes in curvature compared to our water control. And again, we see similar trends for that, for the midpoint and the tail point. Another interesting thing we can do is we can look at the whole body movement. So if we took together the head, the mid, and the tail and averaged those together, we can look at the speed in the black bar and change in curvature in the white bar. Speed is measured by micrometers per second, and again, change in curvature is 1 over micrometers. Kind of pulling out what we've seen in the previous few slides. Again, our 2 treatments show less movement compared to our water control. So that's kind of really saying, hey, our C treatments are kind of doing something with movement. So once we figure out how the nematode's moving, let's figure out what's attracting it or repelling it. And one of the ways, or the way we did this, is using a microfluidic chip. So using that, we can answer the question, do C treatments attract or repel nematodes from the plant? These microfluidic chips, with help from Augie Beeman and the Pandy Lab again, we measure the attraction or repulsion of nematode to a given liquid treatment. Microfluidic filters help with this. So here's 4 different experimental lanes on one chip. Taking a closer look, we have 2 treatment ports where we can put a control in one of them, and then sort of a nematode resting area. Connecting these nematode resting areas are these microfluidic filters. They're about 30 micrometers in width and 5 micrometers in height. That small channel allows for the slow diffusion. Here, these are just color food dyes, over a 24-hour time period. Because the soybean cyst nematode is so small, it takes forever for it to move a given distance. If you were to do this on an agar plate, the chemical treatment would move so far or so fast across the plate, you wouldn't be able to test the differences or measure differences with the nematodes. This allows us to do that because the treatment moves so slow. So we have a couple controls here. Potassium nitrate is a positive control, calcium chloride is a negative control, and water. And we're looking at the proportion of juveniles that have moved either towards or away those controls. And that's calculated by calculating the number of juveniles at a given treatment port divided by the total number of juveniles per that experimental lay. All right? With potassium nitrate, we see strong attraction, which is good. With calcium chloride, we see repulsion, which is good. And with water, we don't see attractiveness at one side or the other, showing that our chips have no bias one side towards the other. Adding in our seed treatment leachates with our control solutions, we measured the same things, but no significant differences of attraction or repulsion amongst those treatments. All right, now that we looked at nematodes moving and nematodes being attracted, let's look at nematodes getting into the roots. That's their whole plan. So a common method to look at nematodes penetrating into roots is to pull the roots out, clear the roots with bleach, and stain the nematodes a nice little pink color. It looks pretty, it takes good pictures, but it's really hard to count all these nematodes in, say, a 20, 30-day-old root because there are so many different planes that you have to adjust the focus in on. So I count 1, 2, 3, 4 nematodes here, and there's one that you can barely see here. I guarantee you there's probably more over here that you don't see. That's really ineffective if you want good data when you're trying to measure the effects of these seed treatments. So I came across another method. Let's macerate it. If you've ever made a smoothie and you blend up some strawberries, the strawberry seeds remain intact. Kind of the same thing with nematodes inside soybean roots. If we blend up the soybean roots and make a smoothie, it's not going to taste good, but we see a lot of these nematodes that we can count. So let's compare the two methods. I grew soybeans in naturally infested soil for 15 days, pulled it out, counted the total number of juveniles to the best of my ability in those roots. White bars. Those same exact roots, I then froze, chopped up, and macerated, made a smoothie, and counted the number of nematodes in that, which just kind of looked like this, black bar. We see about an 80% difference between those two methods. I'm going to stick with this maceration method. So this maceration is much easier, better on the eyes, and more effective than just staining alone. So let's take it one step further. How effective is a protection? How effective is that seed treatment protection at distances away from the seed coat? So let's measure the nematode's ability to penetrate at either 2, 5, or 7 centimeters away from that seed coat. So I jerry-rigged a 50 mil conical tube, cut off the top of the tube with a hacksaw, and then drilled in holes at 2, 5, and 7 centimeters down from the top. I planted seeds, either treated or untreated, in sterile soil within this tube and inoculated juveniles, about 200 of them, at one of those 3 distances. After that, I pulled them out after 24 hours of inoculation, froze it, macerated it, counted the number of nematodes. And the way I'm going to show this data is I related the penetration success to our untreated control, Williams 82. So in this case, I'm going to talk about proportions. And that's just the number of nematodes for a given treatment divided by the number of nematodes for that untreated control. If our proportion is larger than 1, that means that there were more nematodes than our untreated control, or that treatment really didn't work. If it's less than 1, that means we had nematodes less than our untreated control, and our treatment worked. And all of these were just tested against the mean or the null hypothesis that the value was 1. All right. So again, we have our tube. We have our 3 different depths: 2, 5, and 7 centimeters. And we're just counting or looking at the proportion of juveniles per gram of root. So let's look at the top first. First thing we see are 2 treatments, Avicta and Cloriva, have much less, or a decently amount less of nematodes compared to our untreated control. So our treatments worked. Looking at 5 centimeter depth, we still see less nematodes compared to our untreated control. A little bit more than 2 centimeters, but our treatment's still working at 5 centimeters in depth. Looking at 7 centimeters, we just see the Avicta treatment working. Avicta didn't move down that far. So our seed treatments were able to reduce nematode penetration at these 3 measured depths: 2, 5, and 7 centimeters. All right, now the nematodes are in the roots. How are they developing? So, do seed treatments affect the nematode's ability to develop within the plant? If we have arrested or hindered development, perhaps that means we're going to have fewer generations